Okay, so I hope you people have already gone through the previous lecture that ends with this generating and visualizing this electrostatic potential. From today onwards, we are going to start out with a new topic wherein I would introduce you with a few very important concepts in computational biology. Okay, so the very first, to begin with, I would first like to introduce you with the concept of structural alignment. Okay, so you might have heard of sequence alignment. Okay, सीक्वेंस अलाइनमेंट के बारे में आप लोगों ने सुना होगा. Just a minute, someone has. Okay, so you people might already be aware of sequence alignment, wherein you try to align the proteins at the sequence level. Okay, and at the end of such sequence alignment, you might end up concluding that the two provided sequences are similar or are dissimilar. You can also quantify the align. You can also quantify the similarity in terms of percent similarity or percent identity. Okay, so in this topic, what our major focus of discussion would be to align the protein sequences at the structural level rather than sequence level. Okay, so well, say for example, if you have got any protein A and you have got this protein B, and this protein A has got structure like this, and your protein B has also structures that is similar to protein A. Okay, so now you can ask questions. Well, say for example, if protein A is structurally similar to protein B or not. Okay, so that is what the question is needed to be answered, and how do we do that would be the major focus of our interest in today's and next lecture as well. Okay, so we are going to infer the structural similarity between two proteins at structural level. Okay, so now when you want to compare the two proteins at structural level, what you need to do first is that you need to align the two structures first, right? And after alignment, you need to figure out some quantity that would express the similarity or difference between the two protein structures that you have aligned. Okay, so well, say for example, what you would do is that you would first align these two proteins. Okay, well, say for example, this is your protein A, and then you would end up aligning your protein B as well. Okay, this is your protein A and this is your protein B. Okay, and once you align structurally these two proteins A and B, then you need to find out certain quantitative term that would quantify the similarity between the said two proteins when they are aligned with each other, and the quantity that is used to measure the similarity between the structures, we call it as RMSD, or root mean square deviation. So what is this root mean square deviation? This root mean square deviation is one of the parameters that is used to express the difference between two proteins. Okay, or and how these things are done, I would just let you know. Okay, so one more concept that you might also want to learn is that whenever you calculate this RMSD between any two proteins A and B, you can calculate RMSD in or you can say you can calculate this RMSD at different levels. Okay, so we'll say for example, I would just well now let us kind of visualize, or let us kind of create a simple illustration, like we'll say for example, if you have this protein A and B, therefore all of your proteins will usually have backbone atom, okay, and C alpha, then this is your carbonyl carbon, then from here your next amino acid residue would start, okay, then this is C alpha of next amino acid residue, and then there is carbonyl, and the structure would continue so on and so forth. Okay, well now let us assume that at the very first position you have CH3 group over here, and therefore this amino acid residue would be alanine. Okay, and well say for example, you have CH2 over here, and there are again this side group, so this is your, sorry, so you have this CH2, let us have aspartate over here. So, okay, and this is your aspartate. Well say for example, you have a protein A, and at its N terminus we have alanine followed by aspartate, and similarly in protein B we have this C alpha, then again this would be CO, again there would be NH, then again we would be having C alpha carbon atom, then CO, and this would continue. Well say for example, well, let us say we have valine at the first position here, okay, and we'll say for example we have aspartate at second position. Just a minute, someone is about to, okay, so here we have we want to align these two structures, protein A and protein B. These are the two structures that we need to align, okay, and what I have done is that I have drawn the backbone of these two proteins, okay, and we'll say for example the very first amino acid residue in your protein A is alanine, and second amino acid residue is aspartate. On the other hand, similarly I have drawn the backbone of protein B, and the first position in your protein B is valine, and at second position you have got aspartate. Okay, so what we are supposed to do is that we are supposed to find out if these two proteins are similar to each other or not, and in order to find out answers to this question we need to align these two protein structures. And in order to align these two protein structures, what you usually do is that you first superimpose. अलाइन, जब अपन प्रोटीन अलाइन करते हैं when we align the two protein, what we actually do is that we try to superimpose these two protein over each other, and you might end up with something like this after superimposing the structure of these two protein. And after superimposition, you would now calculate the RMSD between the two proteins, and based on whatever value you get for this RMSD term, you would then decide, or you would then conclude if the two proteins are similar or not. Okay, so this is what usually happens in the structural alignment. Okay, so well, say for example, now you can say that the RMSD between these two proteins can be computed in terms of all atoms. Okay, or what we call it as all atom RMSD. Or what you can do is that after you can also compute this RMSD considering only the backbone atom from your protein 1, and you can align it with the backbone atom of protein 2. Okay, so now if you consider only the positioning between two protein molecules, and if you consider only the positioning of backbone atom, you say that you are computing backbone RMSD. Okay, or what you can do is that you can, while aligning the two protein or while superimposing the two protein, and further while you calculate the RMSD, if you consider only the position of C alpha carbon atom between these two protein for calculating your RMSD, you say that you have calculated or you have inferred the structural similarity on the level of C alpha carbon atom, and therefore we say, in such case we say that we have C alpha RMSD. Okay, so whenever you superimpose two protein and you calculate the RMSD between the two protein molecule, based on what is the subset of atom that you are considering for calculating this RMSD between the two protein, you can have all atom RMSD, you can have backbone RMSD, or you can have C alpha RMSD. When we say that we are using all atom RMSD, इसका मतलब क्या है कि after superimposition, while you calculate this RMSD term, you have considered all of the atoms from protein A and protein B for calculating that RMSD. Okay, in that case, you say that you have calculated all atom RMSD. On the other hand, you can limit the RMSD calculation to only the backbone atom. So after superimposition, if you only consider the position of backbone atoms from protein A and position of backbone atoms from protein B, and then compute RMSD, we say that you have calculated backbone RMSD. Or the third level is you can only consider the position of C alpha atom from protein A and its corresponding C alpha atom from position B. In such a way, if that is the case, you say that you have calculated C alpha RMSD. Okay, so there are different levels at which you can, you can compute RMSD between two proteins. Okay, so now the question might arise, when should I use this all atom RMSD? When should I use this backbone RMSD? And when should I use this C alpha carbon RMSD? Okay, so now the answer would be quite simple. You are supposed to use this all atom RMSD when, we'll say for example, you end up comparing conformation of the same protein. Okay, well, say for example, if you are interested in finding out that there are two proteins, well, say for example, if you have got hemoglobin Sometimes hemoglobin, there are few models of hemoglobin that are crystallized when it is bound to oxygen. Okay, and there are certain model of hemoglobin that are crystallized without oxygen. So if such is the case, if you want to find out what is the difference between these two structural state, one is oxygen bound and another one is oxygen unbound, or the structural state of hemoglobin where there is no oxygen, in such a case you might use all atom RMSD. Okay, you can use backbone RMSD when you know that there is significant similarity between two proteins. Okay, well, say for example, if you want to calculate RMSD between, say for example, a DHFR protein from human and from chimpanzee. Okay, now you know that these two proteins might have significant level of similarity between the two structures, and in such case, you might choose backbone RMSD. Okay, so when to use this C alpha carbon atom RMSD? You would use this C alpha carbon atom RMSD in the case when you know that these proteins might not have very similar structure. Okay, so say for example, if these two proteins are quite diverse, or they are placed, they are evolutionarily far distantly related, that means you might have an idea that these protein might have similar fold, but there might be differences. Okay, in such case, you would consider C alpha RMSD. Okay, so I would demonstrate you how these things would work. Okay, so now let us move on to a discussion where I would tell you how these RMSDs are calculated. Okay, so now we people are on the common agreement that these protein structures are generally described in Cartesian coordinates. Okay, so what we have is that for both the proteins, so whatever atoms that are involved in your protein molecule, for each atom we have got respective x, y and z coordinate. So we exactly know where exactly this nitrogen, this C alpha carbon atom, this carbonyl carbon atom, sorry, this carbonyl carbon atom, this carbonyl oxygen atom, the C alpha carbon atom के बाद में ये है C beta carbon atom. Okay, so where, what are the exact location in terms of this x, y and z coordinate that we already know. So those are described in Cartesian coordinates. Okay, so now this RMSD is basically a function of distance between atom in one structure and the same atom in another structure. So RMSD basically क्या है? What is the distance of the C alpha carbon atom from protein A as compared to protein B? Okay, so the comparative distance of analogous atom from protein A and protein B, based on this distance, we would calculate this RMSD. Okay, so this is what it is. So, well, say for example, you have got these two proteins that you are supposed to superimpose and then find out the RMSD between these two proteins. Okay, so one of the protein is protein A and another one is protein B, that is shown in this pink and cyan color or blue color. Okay, so when you compute the RMSD, first thing that you should do is that you need to superimpose these two proteins. Okay, so now if this molecule A or protein A start out in different position with respect to the other molecule in protein B, the RMSD would be high. Why? Because the distance between the corresponding atom would be higher. Okay, I would maybe I would demonstrate you first so that things would become more clear. Okay, so well, let us begin with our simple protein. Okay, what we have over here is the alpha chain of hemoglobin that I am loading, and what we have is this myoglobin that I am loading. Okay, now if I color it by layer, what you can see is that the yellow colored structure is your hemoglobin, alpha chain of hemoglobin, and blue colored structure is your uh, myoglobin. Okay, well, now let me show this by, let me color, by ribbon. Okay, and let us color by chain. Okay, now let me display this. Where is the, okay, so here are the two structures that we are supposed to find out if these two structures are similar to each other or not. So for doing this, as I have already said, that first we need to superimpose these two structures. And for superimposition, we have a simple option over here in terms of this magic fit. So this is where you can decide at what level of what would be the set of atom that you would be using for superimposing the two structures. Okay, either you can use the C alpha carbon atom for superimposition, or you can use this backbone carbon atoms, backbone atoms for superimposition. You can use side chain atoms for superimposition, or you can use all atoms. So I would select C alpha carbon atom in this case. And what you have to do while superimposition is that you need to set one reference model. And what this would mean is that your myoglobin would be superimposed over hemoglobin. So the very first atom that you usually load is by default considered as the reference model. Okay, and when you hit this OK, now you have superimposed these two models. Okay, so now after superimposition, what you can do is that again you can calculate the RMSD between these two protein by simply using this button called as calculate RMS. Okay, so it would again ask you over here that if you want to calculate the RMSD considering only C alpha carbon atom, or if you want to calculate the RMSD considering only backbone atoms, or if you want to calculate the RMSD only considering the side chain atoms, or if you want to calculate the RMSD for all atoms. So if you select this all atoms, that would eventually mean that all of the atoms in these two proteins must be, uh, must be similar, or you can say ki the number of atoms in two proteins should be the same. Then and then you can use this all atom RMSD. Okay, for time being we would be using C alpha carbon atom for calculating the RMSD after it has been superimposed. Okay, well, now when you hit OK, you would find out that the RMSD between the C alpha carbon atom of hemoglobin and myoglobin is supposed to be 2.06 angstrom. Okay, so in this way you can infer the structural similarity between two proteins and quantify it in terms of this RMSD. Okay, so this is what we call it as computing RMSD. This is what we call it as computing, this is what we call it as performing structural superimposition or structural alignment. Okay, so how do we compute RMSD now? The formula for computing RMSD is very simple. So what you usually do is that in order to compute these meaningful RMSD, the two structures under consideration must first be superimposed. That is what we did at the very first step. Okay, then once the two structures are superimposed, then you compute RMSD. Now in order to perform superimposition of any two proteins, first the sequence of those proteins are compared. Okay, superimposition of protein structure usually starts with sequence comparison. Okay, now the objective of performing this sequence comparison is to establish one-to-one relationship between the pair of atoms from which the RMSD is needed to be compared. Okay, so well, let us go back to our this slide. So if you want to superimpose the two proteins, we'll say for example in our case protein A and protein B, in order to superimpose, first you need to align the two sequences. Okay, well, say for example in our case here, protein A and protein B at the very first position, in this protein we have got alanine, and second position we have got aspartate, and in protein B you have got again valine, and at second position again you have got this valine, and at second position you have got this aspartate. Okay, this is your protein A and this is your protein B. So when you compare these two sequence, you need to find out one-to-one relationship between the atoms that are involved in the two proteins that are supposed to be superimposed, and then eventually you need to calculate the RMSD. Okay, so in this way, while you compare these two sequences, you establish one-to-one relationship between the atoms that are needed to be compared between two proteins. Well, say for example, this nitrogen in protein A is analogous to this nitrogen in protein B, and hence when you superimpose these two proteins, you would compare the coordinates of these two proteins or these two atoms. So the idea of aligning the two structures begin with aligning the two sequences of these two structures first. Okay, so the idea, or we'll say for example the logic of aligning the two sequence is to determine which atom from your protein A is to be compared with which atom from protein B. That is what is the ultimate objective of performing the sequence comparison as such. Okay, so thus when you end up with establishing the one-to-one relationship between the pairs of atom. Okay, so next step that is to be done is to superimpose the given set of atoms in an optimal way, so that the RMSD between the two molecules or the two atoms should be the lowest. Okay, so that is what is usually done. So once the optimal superimposition has been done, you say that you have computed the RMSD between the two proteins as such. Okay, I hope things are getting clear now. Well, now what we do for computing such thing is that you first need to establish the one-to-one relationship. Okay, so you might want to figure out ki, well, say for example, if you have got uh, this amino acid residue I in protein A, and the corresponding amino acid residue I in protein B is B. Okay, so what you do is that once you define this one-to-one relationship, what you do is that you calculate the distance between these two atoms. Okay, and how do you calculate the distance? The distance is calculated using a simple equation. Okay, so how do we calculate the distance? Well, say for example, now we know that this atom A and atom B are analogous to each other at position I in protein A and protein B. So since these proteins are described in terms of Cartesian coordinate, we already have the coordinate of this atom AI in protein A. It would be having, say, for example, let us consider it as the x coordinate of atom AI. Then you have got the y coordinate of atom AI, and you have got the z coordinate of atom AI. Okay. Similarly, the corresponding Bi atom would have the x coordinate of atom Bi, the y coordinate of atom Bi, and the z coordinate of atom Bi. Okay, so this is how the two atoms are described in Cartesian coordinate. Okay, now in order to find out the distance between these two atoms, what you do is that you subtract the coordinate of the model from the reference model. Okay, so this is what you've done. You coordinate the model for which the RMSD is supposed to be determined, uska x coordinate. This x coordinate is this number is to be subtracted from the reference model. Okay, so after you perform this subtraction, what you would do, you would take square of it and do the same thing for y coordinate and do the same thing for z coordinate. And after doing that, you would take a square root of the term. Okay, and that square root is called as the distance between the two atoms. Okay, so this is done for a single atom, and therefore you would repeat the same thing for whatever atoms that you are considering for calculating the RMSD. If you are considering only C alpha carbon atom, then you would consider the C alpha residue from next residue pair, that is second residue pair, then third residue pair. And what you would do, you would start out summing up this distance from first residue to the nth residue that is present in your two proteins. Okay, now in order to normalize this, you would divide it by whatever number of residues that are present in your two proteins. Okay, moreover, you might have noted that we have taken the square of the distance. तो ये square इसलिए लेते हैं अपन, if you end up with say some negative value for distance, if you take square, you would have a positive value. And this would make sense because you cannot have negative values for RMSD. Okay, तो ये होने के बाद में what you would do is that you would take the square root and the term that you have calculated is called as RMSD. So this is what is done when you compute RMSD between the two structures. Okay, so are you guys with me, still with me? बात समझ में आ रही है यहाँ तक?
Yes.
Okay, so this is how you compute RMSD. Okay, well now I hope you people are comfortable with this concept. So why? Because we are now going to discuss what are the application of this RMSD. Okay, so now you know that you can answer the question. You can answer the question like if the two proteins are similar or not, or if the two proteins are similar, then what is the level of similarity between them. Okay, now let us try and discuss what or you can say where do we apply this
सर
knowledge. Yes,
सर
बोलो.
सर एक बार वो डिस्टेंस वाली स्लाइड पे जाना सर, इसमें डिस्टेंस कैलकुलेट की थी।
हाँ जी.
यस सर, ये
हाँ
वाली।
हाँ जी.
सर इसमें जहाँ पे डी आई लिखा है
हाँ
उसका स्क्वायर है जो आरएमएसडी वाले में,
हाँ, ये जो डिस्टेंस
जो आपने
अपन
नीचे
नीचे,
डी आई
हाँ,
लिखा है।
उसका स्क्वायर लेते हैं.
और सर आपने स्क्वायर का रीजन दिया था कि मतलब नेगेटिव वैल्यू नहीं होनी चाहिए,
हम्म.
लेकिन सर इस डिस्टेंस में तो ऑलरेडी नेगेटिव वैल्यू वैसे ही हट जाएगी ना, क्योंकि डिस्टेंस के अंदर भी
यहाँ पर
स्क्वायर लिया है कोऑर्डिनेट्स के
स्क्वायर
अंदर।
लिया
यस सर।
है. यस यस यस, बट जस्ट टू मेक
तो
श्योर.
यहाँ पे
हाँ.
सर,
हम्म.
हाँ, यहाँ पे तो ऑलरेडी नेगेटिव वैल्यू हट गई,
वैल्यू
मतलब यहाँ
है,
पे तो नेगेटिव वैल्यू
यही पे हट
कैसे आ
जाएंगी,
सकती है सर अगर
यहाँ
एक्स।
पर ही हट जाएंगी.
हाँ जी। यस सर, तो फिर वहां पे लेने की जरूरत क्या है डी आई में
जरूरत,
जो आपने रीजन।
that is how things have been designed. Okay, these are
ओके।
algorithmic stuff, right? Okay,
ओके।
ठीक है.
ओके सर।
हाँ, so at least you get an idea कि भाई ये RMSD कैसे निकालते रहते हैं अपन. ओके, यहाँ तक समझ में आ गया ना आप लोगों को?
यस सर।
Okay, okay, okay. So now let us start discussing like where do we apply it, right? So there are I'm just going to tell you uh, only three examples where we can implement this RMSD stuff. Okay, so what you can do is that you can use this concept to determine the functional relationship between proteins. For this case, we would be considering hemoglobin, myoglobin, and leg hemoglobin, and how you can derive the functional relationship among these three proteins using the concept of RMSD. Then we would learn how the concept of RMSD is used in validation of your docking protocol. Okay, so when you perform molecular docking, you need to check if the docking algorithm is giving you biologically meaningful results or not. For that case, you usually calculate RMSD. I will let you know in more details of how do we do this. Okay, and the final example that we are going to discuss is all about the convergence test in molecular dynamics. Okay, so when you perform molecular dynamics simulation of any given protein or protein ligand system, or say simple only ligand system, so you would at the end of the simulation you would want to you would want to test if whatever system that I have simulated, if it is conformationally stable or not, if it has attained stability or not. So in that case you would perform this convergence test, and in this convergence test you would also require this RMSD. Okay, so what is time now? Do we still have got time?
Yes, sir. About 10 minutes.
10 मिनट्स।
10 minutes. Okay, ठीक है. तो let us discuss this first topic first. Okay, how RMSD can be used in determining the functional relationship between the two proteins. Okay, so now when it comes to determine the relationship between proteins, there are a few concepts that I think you people should be first aware of before we go on discussing this RMSD and its implication. Okay, so first let me introduce with the concept of homology. Okay, so homology is a term when you use you would use this homology as a term when you would try to establish that any two protein sequences or gene sequences are evolutionarily related to each other. Okay, or you can say the evolutionarily related proteins are called as homologs if they share a common ancestry. If two proteins share a common ancestry, we say that these two proteins are homologous to each other, or they are homologs of each other. Okay, now depending on how they are related to each other, you can classify any two homologs as either orthologs or paralogs, or you can also call them as xenologs. Okay, so one by one, I would first introduce with this concept, and these concepts are very important. We would revisit this concept when we deal with another advanced techniques like homology modeling in your protein structure prediction. Okay, so what are homologs? Homologs, you can call two proteins as homologs of each other, or the two proteins can be called as homologous protein if and only if they share a common ancestor. They have evolved from a common ancestor. Then and then we can say that the two proteins are homologs of each other. Okay, so now, well, let me introduce with the concept of orthology or orthologs. Okay, so well, say for example, to begin with, you usually, well, say for example, you have got some species A. Okay, and this species A contains a gene, or what we call it as, we would call it as ancestral gene. Okay, now in this species, sometimes what happens, there are genetic event like gene duplication. Okay, so what are the results of gene duplication? After, well, say for example, any gene X, if it is duplicated, you would have two copies of the same gene X. Okay, we would call it as copy 1 and we would call it as copy 2. Okay, so now in subsequent stages of mutation, these copy 1 can acquire mutation, and this copy 2 can independently acquire some different mutations. Okay, okay, these two genes can evolve separately by acquiring different mutation. Okay, and after some time, you might have in the same species, you have got say some gene Y and gene Z. After acquiring certain mutation, significant number of mutation, well, this copy 1 can perform function, some function, while this copy 2 can perform certain different function, right? So such two proteins, this protein Y and this protein Z, are called as paralogs of each other. So paralogs are the protein with different function in same species. Okay, well, to give you an example, well, say for example, for a very long time back, in a very long time ago, there might be some ancestral globin gene. Okay, and this ancestral globin gene might have duplicated into two genes. Okay, and these two genes then acquired independent mutations. One of the gene can be called as hemoglobin and another can be called as now myoglobin. Okay, this hemoglobin and myoglobin, after acquiring some specific mutation in the ancestral gene, they have diverged their function, and hence we say that hemoglobin and myoglobin in species human are paralogs to each other. Okay, बात समझ में आ रही? What are paralogs? Paralogs are set of sequences in the same species that perform different function, and these two genes are evolved from say some common ancestral gene. Okay, so such two X and Y genes, in our case hemoglobin and myoglobin, can be called as paralogs of each other. Okay, now this was all about your paralogs, and the key concept over here is that these paralogs arise due to genetic events like gene duplication and mutation. Okay, these paralogs are the two proteins that are present in same species and they perform different function in the same species. Okay, so next what we have is something called as orthologs. तो orthologs के लिए, well, let us consider you have got this species A, and again it has got an ancestral gene. Okay, let us call this ancestral gene as X, and what happens next is that there is an event called as speciation. This species A diverged into two subspecies. Okay, let us call it as a species B and species C. Okay, so the same ancestral gene, due to evolutionary events like speciation, got diverged into two daughter species. Okay, and the same gene X is retained in both the species. The gene X that was present in ancestral species now is present in the daughter species as well. Okay, and this X gene would perform the same function in two distinct independent species. We call such genes as orthologs of each other. Okay, so the protein X in species B is ortholog of protein X in species C. ओके, तो इसका आप एग्जांपल क्या दे सकते हैं? वेल, से फॉर एग्जांपल, again we would stick on to our ancestral globin gene. Okay, and this ancestral globin gene, well, say for example it was present in the ancestor of human and chimpanzee. Okay, and this species A got diverged into species B, that is human, and species B, that is chimpanzee, and it has retained the same hemoglobin in humans as well as in chimpanzee. So therefore we say that hemoglobin in humans and chimpanzee are orthologs of each other. Okay, so the main concept that you should try to remember over here when you consider ortholog is that it is the speciation event that give rise to ortholog. Okay, orthologs are present in different species. Okay, and third is they perform same function, and this is how ortholog differs from your paralogs. Okay, this is how orthologs differ from paralogs. Okay, we must call them as homolog. Why? Because they have appeared from a common ancestral gene. They share same ancestry. Therefore, these are included in homologs. Okay, so by definition, since orthologs have got orthologs have to maintain the same function, it is necessary that they maintains the same structure and hence they maintains the same function. Okay, तो orthologous proteins के sequences और structures में ज़्यादा फर्क नहीं होता. On the other hand, when it comes to paralogs, they have acquired different function. That means you can expect to have a lot of sequence diversity, as well as it is also possible that structures might also diverge. Okay, so in order to maintain the function, it is necessary that it should maintain the structure first, then and then it can maintain the function. Okay, so this is very important point that you should remember, and we would revisit this point when we talk about homology modeling. Okay, good. And the third type of homologs that sometimes appear, they are in form of xenologs. ओके, तो वेल ये एक्सप्लेन करने के लिए, वेल से फॉर एग्जांपल, if you have multiple species that you are comparing with, okay, species A, species B, species C, species D, okay, and if these species are somehow related like this, okay, this is the phylogram of the say two species. Okay, sometimes what happens is that you would end up finding certain genes in a single species, which is the you might end up finding certain genes in this species B, and these genes are not present in its sister taxa. Okay, sister taxa means what? This protein, this species A can be called as sister of this species B. Why? Because they are placed very close to each other. Okay, so sometimes what happens is that suddenly, all of a sudden, you might end up finding certain genes in this species B that are not present in its sister taxa, or that are not present in this entire clade. Okay, and such a type of genes, they show high similarity with say certain viruses or say certain bacteria. ओके, तो ज़्यादातर ये जो सीक्वेंसेस ऐसे होते हैं जो सडनली कोई स्पीशीज में आ जाते हैं, और उनका सिमिलरिटी क्या होता है, किसी वायरस के साथ में ज़्यादातर दिखाई देता है. Such a type of species are termed, or such a type of genes are termed as xenologs, and the xenologs usually are an artifact of the process called as horizontal gene transfer. ओके, तो ये जो जीन्स होते हैं जो अचानक से इवोल्यूशनरी ट्री में किसी स्पीशीज में आ जाता है, जो उसके आजू बाजू के स्पीशीज में आपको कभी नहीं दिखेंगे, वी कॉल दोज एज xenologs. Okay, so well, say for example, now what might be the events that would result in formation of such xenologs? Okay, so well, say for example, uh, well, this species B that we are considering, it might happen in the evolutionary course that at certain point in evolution, this species B, the population of this species B might have been infected by say some virus. Okay, and well, so for example, it might be the similar situation that is happening now. Well, now you know that human population, most of the human population is now infected with your coronavirus. Okay, and the you know the tendency of any virus is to incorporate their genomic content into the host species. Okay, so if most of the individuals in any population acquires certain genes due to viral infection, then that genes, if are found to be favorable for the survival of that species, then they are transferred to next generation or next species, or they can also be repurposed if they are found to be important. Okay, so such genes which are suddenly found in the species B and are similar to certain genes from, say, some virus, these two proteins from the virus and the species are called as xenologues of each other. Okay, so here are the three concepts that we are going to use in our next lecture. Okay, ये समझ में आ गया सब लोगों को?
Yes, sir.
Okay, so I think it's time now, right? So we would discuss this in our next lecture.